Some of you have not been here before, so I would like to pose two quick quizzes. Does everyone know what that is? You don't recognize that? That's the bridge across the San Francisco Bay, right? Everybody agree? Come on. What is it, Alan? It's the Brooklyn Bridge. So this is the most famous bridge in probably bridge history. And it's the logo for this course that we have. Because we are all, like the two gentlemen on the catwalk, crossing between Brooklyn and New York. Life is never the same on either side of a bridge once it's been constructed. And in fact, if Brooklyn, on the other side there, represents advances in basic biological, mathematical, engineering sciences, and over here in New York, we have patients with sicknesses that we'd like to understand and treat. Many of us think that the biggest problem, or a huge problem, in biomedical research is that there aren't enough bridges. And we all speak our own language. We all have our small piece of the pizza pie. So the purpose of this course is to help stimulate particularly young basic Ph.D. scientists, but everybody, in what the problems are and what do we know. So the goal is to excite you, not to fill your heads full of a lot of numbers and so forth. This is not a medical school course that you're going to have a quiz at the end. So I have another question, which I found interesting. Does anyone know the origin and meaning of the word parasite? Myra, do you know? Well, I didn't know either, so I looked it up, and it's really fascinating. Of course, what's underlying our topic of today are parasites. So the meaning of the word parasite, para means to sit opposite. Remember in chemistry, the para position. And site refers in Latin to sharing a meal. So somebody who came and sponged a meal, in other words, they came, they didn't contribute very much, but you fed them. That's the origin of the word parasite. And of course, it's matured greatly. And now we're discussing parasitology, which is the study of how biologic species infect the host, utilize the energy sources of the host, establish some chronic symbiotic relationship, which may eventually, for some diseases, kill the host. And it certainly can contribute to impaired health and survival. And these are the great neglected diseases, which as a topic is an outstanding example, which we'll discuss today, of bridging basic sciences and truly major human health problems. As most of you know, these are the diseases that are most abundant, most chronic, most distributed around the world, particularly in the so-called underdeveloped world, and in their own way are devastating. Now, why are they neglected? What we're going to talk about is a beautiful demonstration that building a bridge requires more than just money. It requires human catalysts who sort of see beyond the curve. So I have to give you a little story. So Ken Warren was actually a student of mine. I was his intern when he was a fourth-year student. I was his resident when he was an intern, and we remained friends throughout his entire life. Now, Ken was a very dynamic person who was trained in parasitology and medicine. And in those days, parasitology was mainly nomenclature, categorizing, the brilliant work done on epidemiology and the extraordinary life cycles and so forth, but mechanistically, in terms of actual cellular processes that were going on, there was a huge gap. It needed a bridge. And Ken, largely through some work that was started here when he was a fellow for a short time, he convinced the Rockefeller Foundation to buy into a project he called the Great Neglected Diseases. And that led to a transformation of tropical and global medicine. So here he is, a very dynamic man. So what did it do? In the course of 10 years, it changed parasitology from taxonomy to what you might call modern science. They provided funding for, I think it's 14 outstanding basic science laboratories, most of which were in immunology. And the only requirement was that people came to an annual meeting and presented real work. And if it wasn't good, they didn't go very far. So he was convinced that non-parasitologists, mainly leading immunologists, could get involved in understanding diseases such as malaria, schisto, worms, et cetera, et cetera. And he convinced them. And those who knew nothing about parasitology, many of them were sent for a short period of time to the London School of Tropical Medicine, where they learned at least the ballpark. The program produced major advances, and it produced what you might call the next generation of leaders in global research. Now, the interesting thing is it was really the product of not just one man's. Ken was the catalyst, but he was also influenced by Franklin Neva, who was here and beloved at the NIH as the director of the Laboratory of Parasitic Diseases for many years, and others. But he carried it through. He had ideas, convictions, imagination, and he had assured funding. You didn't have to write a grant for productive research. And then, as foundations do, they get the ball rolling, and then they roll it to someone else. And the program ended with a new Rockefeller president, but it set the stage, who had other ideas, but it set the stage for work which remains important and exciting, and new approaches to the treatment of the great, truly neglected diseases. Last thing, this paper by another old colleague, Barry Bloom, is, in my view, a classic. And even though it was written in 1979, I would urge any of you who have not read it to read it. Games Parasites Play. It gives you the feeling of the enormity of what happens with different parasites that permit them to continue to maintain this unique relationship with their host. So our speakers today are world-known experts in this exciting field. Alan Sher received his Ph.D. from the University of California in San Diego, did a postdoc in parasitology at the Medical Research Council Laboratory in Mill Hill, London, which was a center for immunology. And in 1980, after having been at Harvard for several years, he came to NIAID as a section chief in the Laboratory of Parasitic Diseases. He's been promoted. He's an NIH distinguished investigator, and his work in particularly immunology, studying host resistance and immune regulation, is widely cited, and I presume that's what he's going to discuss today. Now, our second speaker, Thomas Nutman, got his medical degree at the University of Cincinnati, did medical training at New York University, came here in 1982 as a postdoctoral fellow in the laboratory, which he now heads, the Laboratory of Parasitic Diseases. He is also the director of a NIAID international program in research in India and also in Mali. He's also widely known, recognized, and his contributions, particularly related to helminth infection, are widely cited. So we're very grateful to both of you for being here. And in particular, we're grateful to a young lady who will be interviewed and discuss her illness with you. Okay?
So thank you very much. I'm Tom Nutman, and today I'm just going to have one of our patients who actually has had one of these what I'm now calling, what I'm going to talk about today, one of the neglected tropical diseases, which is part of these great neglected diseases, is going to tell us a little story about how she acquired this infection, and then if and obviously, if there are questions, you feel free to ask of her. But maybe, because I think this is, her story is very analogous to many of the kinds of patients that we see here at the NIH. And as I hope you'll hear at the end, we'll also give you a sense for how we've been able to, by closely observing and treating some of these patients, come up with new and interesting observations that has sort of taken us from the clinic to the lab and then back again. So maybe you could just start by telling us your age and then where you traveled in your profession, and then what happened to you.
Sure. So I am 29 years old. I work for a nonprofit organization that does work in Central African Republic and Democratic Republic of Congo. So I had traveled within 6 months of each other. I took 2 trips, one to Uganda and then Central African Republic, and then the next to Uganda, Central African Republic, and Democratic Republic of Congo. Both of these trips happened one happened in late 2015, and the other one happened in April of 2016. When I returned from my trip in April of 2016, I came back, on a long flight and had the usual, just some general swelling overall, like in my ankles. I didn't really think anything of it. I, went home, slept it off, kind of noticed that it was still there, and then noticed that it was persistent in my left leg, especially my lower left leg, for a period of time. And then one of my friends actually convinced me, you probably should go get that checked out by a doctor just to make sure you don't have a blood clot. So I ended up going to my primary care physician. She had tested me for a blood clot. We did a couple ultrasounds, all came back negative, didn't find anything wrong. My primary care physician just was like, I'm not sure what's happening here, but nothing else is going on. Maybe you just have weak veins. We're not really sure. Let's, monitor this over a period of time. If anything else comes up, please let me know and come back. Let me know if you have any additional symptoms. Nothing else happened from April of 2016 until around late October of 2016, so almost 6 months later, when I started to get some, at the time I didn't know, but now I know it is some really random allergic reactions. So I was just becoming really itchy all over in random places. I was having some redness on my skin. I've never been allergic to anything before, so I wasn't sure what it was kind of just dealing with it. Figured that I developed some type of allergy, but that my doctor wasn't really going to be able to do much for me. So I tried switching all of my products, hypoallergenic products. Nothing was going away. So that lasted for about 6 months until April of 2017, when I was at my office. I was working. My colleagues were around me, and I had felt in my right eye, in the corner of my right eye, I felt like what felt like it was an eyelash stuck there. I felt some irritation, was just like, okay, that's interesting. Went to the bathroom, didn't see anything there. My eye was definitely irritated. It was a little red and bloodshot, but wasn't feeling anything else. Decided that it was probably time for me to take a step away from my computer for a bit. I went out. I went for a drive and I pulled into a parking garage and I was starting to feel something a little different in my eye. It was actually on the inside of my right eye. What I would describe as felt like I can only describe it as like a pulsing or a throbbing sensation. And I was like, that's interesting and feels different. I immediately pulled the mirror down and saw something in my eye, which at the time was in my head. I was like, that's not what you're seeing. You're stressed out. You're totally okay. It looked like a small worm. So it was very visible, was moving in my eye. So my first reaction was, that's not what you're seeing. But then I did a quick Google search of worm in eye, and the first photo that came up matched exactly what I was seeing. I went to the CDC website, and all of the symptoms in the timeline matched. So it had been about a year since I was back from my trip. Everything matched up. So then that is when I decided to return to my doctor and say, I found this thing called Loa Loa. It seems that I have all of the symptoms here. The timeline matches up, and we pretty much went to my primary care doctor and said, I want to be treated for this, or can you do a blood test for this? And then through a series of seeing my primary care physician and then an infectious disease physician, was then referred to NIH.
Can you just describe a little bit more about when you had the worm crawling across your eye? Did you have any other symptoms? Many patients describe significant itchiness or pain. Was any of that part of it?
At that time, with localized to my eye, no, I didn't have anything besides the irritation that I was experiencing earlier, which again, to me, the best way to describe the pain level was like if you have an annoying eyelash stuck in your eye, right? You can't get it out, you don't know where it is. But besides that, not really a lot of itchiness. There was a lot of redness. My eye looked pretty bloodshot from the outside, but you couldn't see anything else. I actually, I was at the grocery store, so I went to the grocery store, went back to my office. By the time I came back and told my colleagues that I had seen, nothing was present in my eye. They were convinced that I did not see a worm in my eye. It wasn't possible. So yeah, there was nothing else associated with that. However, I was still having quite severe for me allergic reaction throughout my body just during that time period. On my arms, I came back with, some bags on I was carrying, and immediately when you put them down, you could see the redness on my arms just from holding the bags.
Okay, and then one last question. We'll cut to the fact that we did treat you over time, but something happened along the way during the treatment. Is that not, you want to maybe tell everybody about that?
Yeah, so I was referred to NIH. We started going through the process of treatment. I was briefed by the doctors very well informed on what the process was, how long treatment was, and the process of what the medicine would do to the worms that were in my body. And so one of the things that I was told was about how the adult worms are killed by the drug that I was taking. And I was told that I wouldn't notice them. They would calcify under my skin. I might have a small bump somewhere. So it's important to note that while I was experiencing the swelling in my left leg, and it was really only in my left leg that I ever experienced it. In recollection, I do remember having a bite there when I came back. That was kind of on my back left thigh. And so that had been kind of an area of focus every time that I returned to NIH to see if the swelling was changing, if it moved anywhere else. And in my first course of treatment, I noticed one day just in my upper thigh that I was feeling some irritation there. And I was like, oh, that's interesting. You know, I went home, you know, didn't really think anything of it. It was during the summer, so it was like hot, sweaty time. So I was just like, you know, maybe I'm just irritated from wearing some tight pants. I was getting changed, and I thought what I saw was a piece of elastic. So that's what, to me, in my brain, I didn't think anything of it. So I just, you know, was like, oh, there's a piece of elastic there, stuck to my skin, pulled it off immediately, was not feeling any more irritation or pain. And then I realized when I had set it down that it was moving. And so then from there, recognized that that was likely a worm. I got in touch with doctors here, was like, is this possible? Could this happen? And it was told it's pretty rare, but it is possible. And then from there was paying a lot of attention to what was happening in that leg as my treatment was happening. And I noticed I had another like small bump. The best way to describe it is it looked like almost like a pimple on my leg. And I accidentally scratched it, and I had another worm that was there as well. Yeah.
Okay, I think that gives you the flavor of at least this infection. Anybody have any quick questions? And we'll just go. All right, thank you very much.
I would defer to you to describe a little bit more in the technical scope. For me, they were like, you know, that long, like maybe an inch or an inch and a half.
Yeah, they're usually 1 to 2 centimeters, very thin, and obviously visible to the human eye. Oh, so yeah, talk about what your exposure.
Yeah. So
I would just repeat the question. The question was what kind of exposure did you have in Central Africa?
Yeah, so I was not in any of the capital cities. My organization works in Eastern Central African Republic. It's very far from Bangui, which is the capital. It is a very remote area. It's considered the bush or the jungle. The same thing in Democratic Republic of Congo. I was in a place called Dungu, which is you know, also, you know, fairly far from the capital. Considered more, it's more populated than where I was in Central African Republic, but I was also in Garamba National Park as well. So, you know, if you know anything about the region, that's a protected park there, but there's, you know, basically just wildlife, a lot of jungle, a lot of forest.
Are there any other questions for? Wait a minute.
I'm sorry. When was the second worm isolated?
What was it like?
It was.
Did we get the diagnosis for the second worm?
We had the diagnosis already. So this is Loa loa.
In the thigh
and
as
it
well?
was an adult Loa loa parasite that came out following treatment that came to the skin, and in the process of dying probably set up a little bit of inflammation that was so superficial that when scratched, it popped up.
Okay, in the thigh also.
Yeah.
Mm-hmm.
Okay, great. I think.
Okay.
All right, great.
Well, thank you.
Thank you very much.
Thank you very, very much for sharing your story with us.
We're going to use our patient as a sort of a lead-in to discussing really neglected tropical diseases as a title. And as we already heard from Dr. Arias earlier, that it really, I think, the modern age really began with this great neglected disease network that's that Ken Warren started, and it lasted really, maybe it lasted 10 years. I think when they really got started, it really was in progress for about 8 years. And the important piece, I think, here was that it spawned the careers of a generation of what I'll call parasite-oriented scientists. At the time, the disease focus was parasitic infections, but it included malaria. And you'll see where that becomes important in a little while. So fast forward to the year 2000 or 1999, and Jeffrey Sachs and a group of the United Nations, convened a big conference, the United Nations, to develop these things called the Millennium Development Goals, or MDGs. And this was a 15-year program to sort of make the world a better place that went from eradicating extreme poverty and hunger to development. And it was supposed to happen all by 2015. If you see there, there were some things that were health-related, improved maternal health and child mortality. But number 6 was to combat HIV/AIDS, malaria, and other diseases, right? And so, as we all know, there was, even before that, a huge effort and dollars given to HIV/AIDS and malaria and tuberculosis. And they just lumped all these other diseases. And this kind of got a number of people bothered. And from these great neglected diseases to the term neglected tropical diseases, or NTDs, happened. And it is true that it is attributed to and popularized by 3 colleagues of ours, Peter Hotez, David Molyneux, and Alan Fennick. And it really grew out of frustration from the use of the term other diseases in this Millennium MDG number 6, because it created very clearly a 2-tier system where HIV and malaria was versus everything else. And it really made public advocacy for these other diseases, these neglected diseases, really impossible. And it really left out these other diseases in most discussions on global health. And so it really turned out to be a marketing strategy, if you will. And it has actually, and I'll give you a couple of reasons why, really driven a huge boost in funding and identity for a lot of people who study these NTDs. And it's not uncommon now to have departments that are in universities and other places that have an NTD focus. So what are these NTDs? So the reason they're neglected is that they are the most prevalent infections of poor people. And up to half of the 2.7 billion people who live on less than $2 a day are affected by these or infected by these. These are non-emerging ancient conditions. They've been around, they're biblical text and a pre-biblical text of many of these conditions. They tend to occur in indigenous populations. And the other thing that they don't do in large part is they don't kill. They're chronic disabling conditions that cause growth delays, blindness, social stigma, disfigurement. Shown on the right there is a patient with lymphatic filariasis, and you can see the massive elephantiasis in that patient as part of this is taken in India as part of a hygiene campaign to treat this. And we know now from a lot of socioeconomic studies that these are poverty-promoting conditions, driving poor pregnancy outcome, productive capacity, child development, and education. So when you lump all of these neglected tropical diseases together in one sort of moniker, and then you look at the 10 leading causes of life years lost to disability and premature death, you can see that these NTDs actually fall above malaria, above TB, and below some of these others. But suffice it to say, it makes a very powerful argument that actually dealing with these infections actually will lead to improved quality of life. So what are they? So it turned out that, as you can imagine, everybody wanted a piece of the NTD puzzle or a portfolio. And it started out with 13 or 14 sort of targeted infections, and now has expanded to include 18 or 20 different conditions that are caused by protozoa, which are single-cell parasitic organisms, viruses such as rabies and dengue and chikungunya, some of the bacteria, particularly leprosy and yaws and trachoma and Buruli ulcer, a whole bunch of these helminth infections, and I'll get to those in a little bit more detail in a sec. And then recently added on to these are scabies, some of the deep mycoses, and even snake bite snake bites as a serious problem in the tropical world. So our focus here at the NIH, or my group's focus, are these helminth infections or worm infections. It's a fancy word for worm, really. And you can see they get categorized into cestodes and nematodes and trematodes. And you can see the major species that affect humans and how many people are infected with each of these. And just suffice it to say that our group has focused largely on these filarial infections that are up on your left, in the middle of the left, and two soil-transmitted helminth infections. And really what I'm going to just focus in on today is a little bit of our work on worm infections, and particularly the filarial infections. So among these neglected tropical infections are these filarial infections of humans. Shown here are the 8 species of filarial parasites that infect humans, and the numbers of people infected. The top 4, W. bancrofti, 2 Brugia species, Onchocerca volvulus, and Loa loa, are considered the major human pathogens. And I need to point out that collectively they're the second leading cause of disability worldwide, these 4 together. And then thirdly, it's important to note that with the exception of one species of Brugia, all of these have only humans as their reservoir. So these are vector-borne diseases, and so pretty much everything that we have to do to study these infections have to be done in human populations. So they're neglected by everybody but heavy metal bands in Germany primarily. But that being said, they have made it. They've made it to the big time, as shown by these album covers. And what we're going to focus in on today is on the upper right-hand corner, Loa loa. So what is Loa loa? So this is what our patient today had, and I think it's a great example of having paid very close attention to the clinical manifestations of this infection has allowed us to actually learn a lot about the sort of basic science surrounding this, and also to then use that basic science for therapeutic good. And that's the kind of story I'm going to try to tell today. So Loa loa is caused by the parasite loiasis is called by the parasite Loa loa. It's transmitted by a deer fly. I'll show you a picture of that in a second. It has a little bit of a complicated life cycle, but the microfilaria, that's the stage that actually passes from the human to the fly and then back to the human again, is called a microfilaria. That is not the adult worm. The adult worm lives in the subcutaneous tissue largely and migrates. It has a geographic distribution that's primarily Central Africa, a little bit in West Africa. It has a prevalence somewhere between 13 and 15 million people. And as I mentioned, only humans are infected. So this is just a geographic distribution map of Loa loa infection. And the brighter the orange or brown color, the more intense the infection, or the more prevalent the infection is in these various countries. And you can see both CAR and DRC, where our patient today acquired this infection, are really 2 of the most highly endemic countries for this infection. This is the life cycle. This is the Chrysops deer fly that actually transmits the infection. And suffice it to say that the fly picks up the microfilaria in a blood meal. Over a 2-week period or so, they develop into those L3s. That's the bottom one. That's the infective stage of the parasite, and then those are transmitted to another human, and then they develop. And it takes somewhere between probably 6 to 12 months before the adults, males and females, mate and produce microfilaria. So from the time you're infected to the time you actually get symptomatic, it can often be a year or 18 months. And so it's important to know as physicians or looking at patients that the time of travel to the time of symptomatology often takes a good significant period of time, and that has to do with the parasite development. So the clinical manifestations of Loa Loa are depicted here, and interestingly, and this is true of most parasitic infections, that the largest number of people who are truly infected have a clinically asymptomatic condition or a subclinical condition. And when you get pathology, it's either non-specific, and in the case of Loa Loa, you get itchiness, as our patient had today, urticaria, hives, occasionally myalgias. And the two classic symptoms that sort of make us understand what this is are something called calabar swellings and this eye worm. And the other piece to remember is that you can, because of the host's response to these parasites, you can end up getting significant complications, things like endomyocardial fibrosis, which is a fibrosis of the heart, renal disease, CNS involvement with encephalopathy, or entrapment neuropathy or peripheral neuropathy, much like carpal tunnel. So those are complications, and that's the reason that we actually treat people, because you could argue, well, you have this asymptomatic condition, why bother, right? I mean, it's not doing too much harm, apart from an occasional worm across your eye. But it's because to prevent some of these significant complications that we want to treat, and we want to treat as early as possible. So this is what a calabar swelling looks like. These are areas of evanescent migratory angioedema that typically can occur anywhere on the body. It's hard to photograph sometimes, but I think you can appreciate the top of the hand on the right there being quite swollen, or you can see the wrist on the other side and on your left that also is this swelling. It often will come and last for 3 or 4 days and then migrate to another area, and we know that this is probably related to the adult parasites moving across the subcutaneous tissue and leaving its antigens behind, and our body having, let's call it, an allergic type reaction to those antigens. The thing that everybody remembers and was described beautifully by our patient today is the eye worm. And it is not in the eye per se. Well, it is, but it's actually subconjunctival. So just under the conjunctiva of your eye, and it moves. So you can think about this as the same plane as the think of it as the skin of the eye. And so probably it's just like it's moving in our subcutaneous tissue. Every now and then it crosses the conjunctiva, and this is really kind of the hallmark of the infection, and obviously what most people remember about it. Loiasis is also called the African eyeworm, and you can imagine why. This is actually how oftentimes patients get referred to us here in this country. This is a Cameroonian taxi driver in Washington, D.C., and made his way to the ER at Georgetown and was told he didn't have anything to worry about. And then he insisted. He said, I know, I'm from Cameroon, and I have a LOA, and they called the ophthalmologist, and they popped it out. This isn't curative because just like our own patient today, for every one that you happen to see, there's probably many more lying elsewhere. So one of the beautiful things about the NIH and our clinical program here is that it has given us the opportunity to be referred patients with these infections. When I first started here, I got referred a good number of people who were primarily Peace Corps volunteers and photojournalists and missionaries who presented with Loa Loa. And what was interesting to me was that it really wasn't anything like what I had read in the textbooks about the manifestation of Loa Loa. And so we actually made a decision to sort of compare people who are born and bred and lived most of their life in an endemic area with Loa Loa to those who actually acquired it during travel, and compared their just standard symptoms. And what you can see is in our first set of patients, when we're comparing patients that we saw here to patients that we studied in Benin in West Africa, that the likelihood of having calabar swellings was much greater in the travelers than it was in those born to endemic regions. And similarly, those people with relatively asymptomatic infections, no symptoms whatsoever, was really just the opposite. So patients who traveled, and now we have a sense that this has to do with the relative acuteness of their exposure or where they are in the evolution of the infection, were much more likely to have a significant clinical reactivity to the parasite. So we were able, with a fellow in the lab just recently, to pull our much greater experience of only patients that we saw here at the NIH. And a lot of our original observations were found to be quite similar. Calabar swelling much more frequent in the travelers. Both eyeworm and the asymptomatic microfilaria positive patients, much more common in those born in endemic regions. And then the rest was pretty much similar. And if you think about the complications associated with this, they were pretty much identical between the two groups. So one of the things that has sort of been nice about being able to study some of these patients in depth is that we were able to begin to understand some of the pathogenesis of some of the complications. And this is actually a biopsy of a patient with endomyocardial fibrosis secondary to Loa here. And what you can see on the right in the green there is the heart muscle on this biopsy that's actually stained by eosinophil granule protein. So these parasites are very capable, and you'll hear this from Dr. Sher in the next talk, really driving a response that is called sort of a Th2 type response. And in this situation, this Th2 response drove eosinophils into the heart. They got activated. They dumped their granular proteins on the heart muscle and actually induced, we think, fibrosis. So one of the other things that allowed us to do was to study patients sort of longitudinally and come up with this kind of sort of understanding of how the parasites set up an immune response, what the nature of that immune response is, and then what happens over time. And I won't dwell on this because I think Alan's going to talk about this in much more great detail. But suffice it to say that very early on in the infection, from 0 to 6 months or 0 to a year, there is a marked induction of things that are associated with worm infections, things like eosinophils and IgE and IgG4. And then over time, many of these responses get dampened. Being able to study patients over time has kind of allowed us to sort of fill in that graph I just showed you was a graph that kind of was circa, let's say, 1995 or 2000. And now we can fill in many more details. And I'm not going to go into this at all, but suffice it to say that it takes a very long period of time, decades, to dampen down the inflammatory response such that you end up with a relative asymptomatic condition associated with this infection. So with all this sort of data together, we understand that when the parasite and the host, in the context of these filarial infections, the normal response is to have let's say, a moderated or a suppressed immune response. And as a consequence, you end up with this asymptomatic subclinical infection. And if you fail to modulate that response, you end up with inflammation. And that inflammation drives immune-mediated pathology. So in the case of Loa, this calabar swelling. In the case of onchocerciasis, blindness or skin disease. In the case of lymphatic filariasis, or other filarial infections, things like elephantiasis and hydrocele. And we know that that a lot of this is actually the suppression is driven by things like IL-10 and other immunosuppressive cytokines. So we don't just sort of study patients. We obviously want to treat them, and there currently are 2 drugs that we use, and the drug of choice is something called diethylcarbamazine, or DEC. We don't understand its mechanism of action, but we know that it kills both the adults and the microfilaria. And like most of these filarial infections, there are post-treatment, oftentimes severe side effects in patients with high levels of circulating parasites, because the drug is so effective at killing the microfilaria that they rapidly kill the microfilaria, they release all their antigens, they drive inflammation, and people can have severe side effects. And particularly in the case of Loa Loa, we're very concerned about central nervous system problems. Ivermectin, which is a drug that recently won the Nobel Prize, or the discovery of which won the Nobel Prize a couple years ago, is also can be used in Loa Loa, but it's only microfilaria soluble. And because of that, it's also associated with severe side effects. So what do you do in patients who are have high levels of these microfilaria? The only drugs that you have are associated with serious side effects, and you want to treat people safely. So one of the things that happens, and I'll get to that other point in a second, and this is what was described by our patient today, is following treatment, you end up with occasionally post-treatment this vermiform eruption on the skin, and this actually is a patient that was an inpatient here at the NIH that we actually just nicked and pulled the worm out. So to prevent some of these sort of effects, we sometimes treat patients with corticosteroids. And one of the things that we developed here was apheresis. So we can transiently, using blood banking material, remove the microfilaria such that we lower the levels to the point where when we can treat patients, we can do it safely. So over the past many years, we've actually done this in 46 heavily microfilariemic patients, and you can see that these levels go down following apheresis. And the point here is that we actually have to do them when the parasites are in the blood. And one of the fascinating things about most of these filarial parasites is its periodicity. And if you just look at the upper right here in corner where it says Loa Loa or LL, this is actually, if you follow a single person and count their microfilaria during the 24-hour period from midnight to midday, and then midnight again, you can see that in the case of Loa Loa, at 12 noon on each of the successive days, the parasites are in the bloodstream. And this periodicity, we're beginning to get an understanding of what drives it, because as you'll see in a second, now we have the genomes of these, and we can begin to sort of understand that. So when you do these apheresis of the patients, this is actually looking at just a picture of from a cell phone on think of it as the blood pack that you get from the blood banking machine. You can see the parasites just wiggling in the blood. So we can actually then use this to our advantage, and we can actually just purify them. Sorry, purify them. It's not going to wiggle, sorry. And we actually then did this from several patients where we purified all of these wiggling microfilaria. Sorry, it's not moving, Mac to PC problem. And we were able then to make sufficient DNA to actually do the genome. And so from this sort of absolutely kind of clinically driven need to reduce the microfilaria, we were able to actually collect enough parasites to, at the time, complete the Loa Loa genome. And so with the remaining little bit of the time that I have, I'm going to tell you a little bit of story of how we've utilized that to get to something of therapeutic value. So when we did the genome, we looked across the genome, and all of a sudden we saw that the parasite contained a set of enzymes, kinases, that are already targeted of FDA-approved drugs. And what's shown in the red there is the tyrosine kinase or Abel-like kinase that are expressed by all the filarial parasites in single copy. And the target of that is a drug called, among them is a drug called imatinib, which has actually revolutionized the treatment of things like chronic myelogenous leukemia. So Elise O'Connell, who was at the time a fellow in the lab, took this on, and first we were able to show that the LOA kinase, as predicted by the genome, structurally was identical to the catalytic subunit of the human BCR-ABL, the ABL-like kinase. And if you look at on the right where the overlap is, that you can see that they basically overlap with an unbelievably high degree of similarity. And suggested that we could actually use imatinib, which is a very safe orally delivered drug, to perhaps treat LOA. So actually, this is a picture that in a second the movie will start. So shown here are the LOA. These are actually a related parasite. Sorry, it's going to go. So shown at the top is in the absence of the drug, and shown at the bottom at the same time, you can see a little bit of wiggling, a moribund parasite in the presence of the drug. And then finally, we were able to use antibodies to actually localize in the parasite where this enzyme is expressed, and largely it's expressed in the female reproductive tract of the filaria. What you're shown here is a worm, an adult worm in cross-section with that little red thing that you can see across separates one of the parasite uterus from the other, and it's where the microfilaria live in the adult female. And we could show that this filarial c-ABL, the target of this imatinib, was strongly expressed in the reproductive tract of the filaria and these microfilaria in particular. So to sort of finish up, what we did was we proposed that we should actually think about getting a clinical protocol to use as a single, to use as an oral drug to actually reduce these microfilaria to levels that were safe then for us to treat with another drug. And shown, and the reason is that these severe adverse reactions is absolutely related to the LOA microfilaria count and then the rapidity with which it decreases following treatment. And so shown in that little cartoon over there is the danger zone, so anything over about 20,000 microfilaria per mil. And if you give ivermectin, or one of the microfilaricidal effects, you could see that you actually drop the level to almost zero within 24 to 48 hours. And the idea was, could we use this other drug, imatinib in this case, to lower the levels down a little bit more slowly, but nevertheless to a point safely where we could then now treat with this. And without giving too much away, our patient today actually was willing to actually, prior to her being definitively treated in a obviously consensual way, to try a single dose of this drug to see what happened. And what you can see here is that over a 7-day period, with a single oral drug associated with essentially no side effects at the doses that were used, that you actually lowered the level, not from 20,000 to zero, but from where she started at 2,200 or so, down to less than 100 or so. And it's actually now allowing us to do a randomized control study for this in Cameroon, where obviously this high level of microfilaria is a problem. So I hope I convinced you that that these neglected tropical diseases, the study of these are actually both important and also able very clearly to be a model to sort of go from the clinic to the bench and then back again, and how I think places that allow for very close observation of the patient and a sort of a marriage between good clinical care and at the same time, let's call it adjunct sort of basic science, sort of gets you to where you want, and maybe a paradigm for many of these other infections. This is not my car, by the way. And before I end, I just want to, make the case that this is that all of science and medicine is sort of a team sport. Obviously, I want to thank the many, many patients, both here and abroad, that have contributed to sort of our understanding, many of my colleagues in all these places, my current lab, many of whom have shown up here today, which I appreciate. And I want to just point out Elise O'Connell, who's in the front row on the right of my lab group, who actually did a lot of the sort of work on the tyrosine kinase inhibitors and its use for what I told you today. So with that, I'll leave you.
Thank you very much, Tom. That was really very exciting. Time for questions. Anybody, please don't hesitate. If you have a question, don't. Go ahead.
So regarding the allergy symptoms,
Yeah.
So do you think this is all from the products from the worms per se, or actually the worm infection can sensitize the immune system, then the immune cells respond more sensitively to the environmental stimuli?
Right. So I think that the so I think the effector phase of the allergy is obviously host derived, and much of this is we think is IgE mediated. So and as is much of the allergic reaction allergic reactions are. We think that the parasites themselves, the parasite antigens themselves, actually drive both a Th2 type environment, but that Th2 environment actually also induces parasite-specific IgE, and that parasite IgE actually mediates on human mast cells and eosinophils and other cells a lot of the allergic reactivity that actually occurs. And it's one of the things that we're hoping, we're actually trying to understand the interrelationship between helminth infections and allergic diseases because with chronic, long-standing helminth infections, it appears that the allergic symptoms are actually downregulated. But what we think is actually happening, at least when you get allergic symptoms, actually is that it is the parasites driving that allergic reactivity.
So while people are, let me ask you a question. On your last slide there, the numbers on the ordinate, where in the patient it comes way, way down. What is that ordinate? It's so many filaria
so
per
it's
what?
microfilaria per mil, and
per
so
mil.
per mil, right? So
there are 2,000.
So 2,000 in our case, we would think of those as a relatively modest number. In studies that I didn't talk about today, where in places like in Cameroon, for example, where we work, it is not uncommon to see patients with hundreds of thousands of microfilaria per mil of blood. I mean, it happens about 2% of microfilaria positive patients, but you can get levels that exceed 200,000 or 300,000 per mil of blood.
Are they asymptomatic too?
So it's really interesting. So the higher the burden of infection, the more likely they are to be asymptomatic. And we actually think that it's actually with the exception of the fact that they end up being the reservoir for continued transmission, that probably it's teaching us something about how the parasites actually downregulate or suppress the immune system. And it also suggests that there are probably we know that there actually are parasite-encoded products that may actually be used therapeutically to dampen either autoimmune diseases or other things, but that's still, I'd say, a work in progress.
So do other parasites within this big family have BCR-ABL?
So they do. So the filarial parasites all have quite a similar, almost identical tyrosine-like kinase. And then the related helminths, such as the schistosomes or at least the trematodes, have a similar not quite as, it's a little less homologous to the human BCR-ABL than are the filarial ones, but it's in the 60% range of identity at the amino acid level. So it is a potential you know, not pan-parasite, but at least targeted parasite directed approach. And we're doing this with some of the other, so imatinib is much safer in a sense than some of these other drugs that target slightly different kinases. And so there's always this therapeutic you know, window where the benefit and the side effects have to kind of be sort of balanced.
What's BCR-ABL doing in the parasite?
Well, so we don't think it has anything. So just remember, the BCR-ABL in CML is actually a fusion protein between a kinase encoded on you know, on one side of the Philadelphia chromosome and you know, and fused to a
That was
to a
my
second.
next question.
Right.
Do they have a Philadelphia
So no, they
chromosome?
do not. They this is probably and probably just like the homologous tyrosine kinases in humans that are not constitutively active, that these are probably have an integral role in the sort of metabolism of the cells of the parasite. So it's the fact that it's the only issue really had to do with the fact that the BCR-ABL, you know, was used as to discover essentially these class of drugs that have been hugely successful, but we're leveraging largely the pharmaceutical industry in a sense for having done all of the sort of safety and you know, safety sorts of and sort of structure function relationships that allowed us then to sort of figure out that we could probably go for this. The issue, of course, then becomes cost, right? So we're talking about, you know, things. So right now, imatinib is even now that it's off patent, is actually expensive in most places, but it's a single dose, so it's not lifelong as is in CML. And the other thing is that it's can be it actually is now listed as one of the WHO essential drugs. Now they're not thinking about giving it out to people with LOA, you know, all over the world, but the point is that it's actually subsidized to a point where it actually can be given at relatively inexpensively to anybody in the world.
So last year, Tom, one of our speakers was Jonathan Slayman, who is the director of the United States Geologic Survey. They do more than make maps. They survey the effect of global warming on the migration of various vectors. And one of the vectors that's on his list is the deer fly. Mm-hmm. And my understanding is, I may be in error from this, but that the deer fly, like other fly vectors, has now, you know, has been detected, particularly in southern states and so forth.
So we have chrysops here in Maryland. We don't have the right species. So you need two things.
So what would it take to have endogenous Loa in Maryland?
Well, it would take a huge reservoir of patients, which is probably not, you know, is not going to happen. It also actually takes the right chrysops species. The ones that we have on the here in Maryland, for example, are not capable of transmitting it. So there is a vector species specificity for both, you know, for the vector and the particular parasite. So yeah.
Any questions? Well, we'll have time after Dr. Sher is finished. Thank you very much.
Thank you.
Very exciting.
I'd like to begin by thanking Erwin for inviting both Tom and I to speak today. I've been in the audience like you people on many occasions and thoroughly enjoyed this whole series, and it's nice to be part of it on the other side. So anyway, I'm going to sort of do a now for something completely different switch here and talk about more of the basic side of work in parasitology, and in particular the immune response to parasites. And the basic premise of my talk is shown here, and it may be a little bit arrogant, I don't know, and that is that really by studying the immune response to parasites, which is something which is spurred on by the quest to make vaccines against these organisms, this has provided fundamental insight into the nature of both the innate and adaptive components of the immune system. So parasitology, or the study of the immune response to parasites, has told us a lot about immunology. And I'd like to go further and say that this represents a major contribution of parasitology to modern biomedical science. Pretty arrogant, I think. So the field that I work in, and actually a number of people in LPD do, including Tom, is a field called immunoparasitology. And I have to say that when I started out in the field, this word did not exist. It was sort of cobbled together by some people. But the definition is very, I think, insightful, and that is, it's a branch of immunology that deals with animal parasites and their hosts. It's not part of parasitology. It's part of immunology. And that's going to be kind of the angle that I'm going to be presenting to you today. So, how did immunoparasitology evolve? And I'm going to give you a little bit of history here. It really came from two different forces coming together. And the first was something called the immunobiological revolution, a great explosion in immunologic research that occurred in the 60s and 70s. And this slide just lists some of the key developments. Demonstration that lymphocytes are key mediators of adaptive immunity, the identification of B and T lymphocytes and their cooperation in generation of humoral immunity and antibody responses, and then importantly, the development of monoclonal antibodies and T-cell lines and clones, which enabled us to identify targets of the immune response. And then this one, which I'm particularly fond of, which is the development of inbred and congenic mouse strains and the discovery of MHC restriction, which enabled us to develop phenomenal immunologic models to study disease. So this was the immunobiological revolution. But simultaneously with this was an enormous explosion in the use of vaccines and vaccinology. And you all know this pretty well. But this slide always amazes me. And if you ever have anybody who doesn't believe in vaccines or is anti-vaccine, show them this slide. So this is the impact of vaccination on all of these major human infections. And you just have to go over to the far right here and see the enormous impact on the prevalence of these diseases that vaccines have created in human health. It's a fact that I think is often overlooked. So at this particular time in history, there was a lot of optimism, and in fact, there was this very famous quote from William Stewart, who was the Surgeon General of the United States in 1965 through 1969. And he is said to have said this, but there's a lot of controversy as to whether he really used these words. And he said, "It is time to close the book on infectious disease. The war against pestilence is over." Now, we all know in retrospect, this was a very foolish statement, because a few years later, HIV came around the corner and blew everybody away. But the point is, this reflects the optimism at the time. And Stewart was no fool and an important man, because he also was very much involved in the beginnings of the anti-smoking campaign in the United States. So he was an important guy. So maybe he said this, maybe he didn't. It's the first example of fake news. Anyway, you could say this if you wanted to, based on the data that I just showed you, the infectious disease and vaccination data. But one area where you couldn't say it was in the area of parasitic diseases, and this has already been explained to you or introduced by both Erwin and Tom, and this idea of great neglected diseases of mankind, parasitic infections. And And this is an old slide, and the prevalence of these diseases has indeed gone down. But I think the key thing is over again on the right, is that the control measures that were available at the time and pretty much still are today are vector control, chemotherapy, and hygiene. And at this particular period in history that we're talking about, there were no vaccines, and as you'll see, they really, for all practical purposes, aren't today either, which is the sad part of this story. So at the time, I was a young graduate student and trying to decide what to do with myself for my postdoc, and I wanted to get involved in something that was more translational. I had more translational import for human health, and with several of my colleagues who are shown here, who are fellow immunologists. Now there will be one quiz in this lecture, which is to figure out which one of these four people is me. I can tell you it's not this guy here because well, I can't use the pointer, but it's not this guy because he was one of Tom Nutman's mentors at Brown University. So that leaves three. That's Paul Knopf. But anyway, we were all immunologists looking for something to work on that would be something that where we would really be needed, and this field of parasitic disease was brought to our attention, and all four of us went into it and working on a disease called schistosomiasis, which Erwin actually used to work on years ago. So so that was our crusade. And really, this highlights the goal-oriented part of immunoparasitological research, which is part of its origin, which is to elucidate mechanisms of host resistance and immunopathology in parasitic infection so that we can make vaccines and other forms of immunologic intervention in human. And I added veterinary disease just to emphasize to all of you that parasites are very important causes of veterinary disease, and that the two are that both quests can be linked at certain levels. So we went to work on schistosomiasis, trying to understand this very complicated organism. But about 10 or 15 years later, there was an enormous breakthrough in the field that kind of had the kernels of everything you wanted from what I just described. And this is work of Ruth and Victor Nussensweig, who are the sweethearts of the field, as you can see here. And they were both, these are both scientists working on malaria. Ruth is a malariologist, Victor more of a basic immunologist. And what they showed, it was really based on Ruth's work. And I should mention again, this is another set of mentors for Tom Nutman. Ruth was, at any rate. And what she showed was that there's an infective stage of malaria called the sporozoite. And then if you immunize mice with irradiated sporozoites, you could protect them against challenge infection with either sporozoites or even infected mosquitoes. And she went on to show that this was mediated by antibodies, and actually made what was then a new kind of tool, used a new tool, which was monoclonal antibodies, to show that a monoclonal antibody could transfer protection to the disease. And this monoclonal antibody recognized a series of repeats on this molecule, which is called CS, and they then cloned the antigen, which again was a technical feat at the time, to clone and express an antigen, and they did so, and were able to use this antigen to, or parts of it, even to immunize mice and get protection and develop an experimental vaccine against malaria. And this, of course, was, I mean, it was very exciting, and it was sort of the latest technology. But unfortunately, these early experiments turned out not to go very far because, and I should say that this strategy of using monoclonal antibodies to identify antigens, cloning them and expressing them, became kind of like a factory effort. Every lab started doing this with their particular bug, and with the idea that they could simply take the antigens that they got, immunize, and you'll have a vaccine against the disease that you want. So, when this was tried with other host parasite models, such as schistosomes, which I was working on at the time, it didn't really work well at all. And even in the malaria system, protection by defined antigen in mice was much less than that conferred by the live intact irradiated parasites. For some reason, the irradiated organisms stimulated much better immunity. And then when they went into humans, of course, there was very poor translation in vaccine trials. However, as we'll see at the end of the lecture, there's some recent encouragement with a kind of new version of this type of vaccine called RTSS. So the question then, what we were left with was how to design effective vaccines. And I've always felt that this should be the strategy, which is to identify the mechanism by which the immune system can control a parasitic infection, and then learn how to trigger those immune responses at the correct level and maintain them in a form that can be recalled upon parasite challenge. That gives you memory, so you can have a vaccine that will last for a long time and doesn't require constant boosting by medical workers. So, so here one confronts something about parasites that's already been noted, I think, by both speakers, and that is that that parasites are really very sophisticated eukaryotic infectious agents. They're complicated, and they've co-evolved with the immune system, and they're very good at evading host effector mechanisms and regulating immunopathology in order to produce long-lasting chronic infections. Their goal is to not to kill you, but to keep you alive so they can transmit themselves to the next host. And this is, Erwin referred to this famous article by Barry Bloom called Games Parasites Play, and this is part of this gaming strategy. So, but there's that's the negative side. On the positive side, though, parasites, in common with some other persistent pathogens, are really venerable scholars of the immune response, and unlike us, are undaunted by its complexity. So parasites have learned to manipulate the immune system. They are good immunologists, and that's why by studying them, I think we can learn a lot about the immune system. So, and they seem to be particularly good at two things, and I call them effector choice. And what that means is what directs the immune system to produce different classes of immune responses. Not all immune responses are the same. We have antibodies, we have T-cell mediated immunity, we have different flavors of immune responses, as I'll explain in a minute. And parasites seem to really know how to direct effector choice. And the other thing that they're very good at is immune regulation. And you heard a little bit about this in Tom's lecture in the case of human disease. And that's how effector responses are controlled while mediating host protection. And the problem here is that you can induce an immune response, but there's always the possibility that in addition to causing protection, it's going to cause inflammation and disease. And parasites seem to have learned how to figure that out so that they keep the host alive and they can transmit their cycles. So, the place where all of this begins is in this what's pointed out here. This is the ultimate effector choice I think, in the infectious disease world, and it occurs in parasites. And this is something that I confronted when I started out in this field, and that is that we really have two basic flavors of organisms in parasitology, helminths and protozoa, and it turns out that the immune response to them is really quite different. So as you've heard from Tom, helminths are famous for their ability to induce responses that are classically associated with allergy. This would be immediate hypersensitivity, IgE, eosinophils, and mastocytosis. In contrast, most protozoan infections, and in particular intracellular protozoan infections, are famous for stimulating cell-mediated immunity, delayed hypersensitivity, and lymphocyte proliferation. And these are in the case of the protozoa, not unlike some of the responses stimulated by bacteria, in particular intracellular bacteria. And this was always a fascinating distinction, and it became even more interesting when it was found out because of the immunologic revolution and the analysis of the role of T cells that both of these responses were highly dependent on CD4 T cells, which are called helper cells, and they are and CD4 T cells, as you remember, are the cells that are critical in host defense and why AIDS patients become susceptible to many infections. So so this was really interesting. But when we thought about it at the time, and it just seemed strange that you could have one T cell that was mediating both of these effects. And it wasn't till later on in the 1980s. So here's the question: how can the CD4 T lymphocyte generate such divergent immunologic outcomes? And the answer came from some very important work by these two gentlemen, Tim Mossman and Bob Kaufman, in the late 80s. And they showed actually that there is not just one CD4 T cell, but there's at least two. And they called these Th1 and Th2. And they were distinguished importantly by their production of cytokines, which are immunologic mediators. And the Th1 cells made interferon gamma and IL-2, two very important cytokines, whereas the Th2 cells produce IL-4, IL-5, and IL-13. And this is all based on very elegant work with T cell clones. And those responses, in turn, were associated with classical cell-mediated immunity and allergy. And so you can imagine that this right away provided an explanation for this dichotomy that we saw in parasitic infection. And we were able to, we and other labs were working with Mossman and Kaufman, were able to demonstrate that in fact this was the case, that helminths stimulate these particular Th2 type CD4 T cells, whereas intracellular protozoa a Th1 type response. So this is effector choice, and the concept has been extended over the years, and it turns out there's more than one type of T helper, or more than two types of helper cells, where we now have at least 6 or 7 of them. That's probably a low ball estimate, and they're all based on their cytokine secretion patterns, and very often they're associated with the response to particular types of organisms. So, for example, Th17 cells seem to be prominent in the response to extracellular bacteria and fungi, and in almost the same way that Th2 cells favor the response to worms and Th1 intracellular pathogens. So the concept really started with parasites and has been extended to other types of pathogens and other types of T cell responses. So so what we've done in the lab over the years is to address a number of central questions about this. We've focused largely on the Th1, Th2 response difference. And so we want to know what are the functions of these responses in host resistance and disease, particularly in parasitic infection. What are the host signaling events that dictate Th1 versus Th2 outcome? What signals occur within immune cells that drive them towards one or the other outcome? And then what are the parasite triggers of Th1 versus Th2 differentiation, and how are they recognized by the host? So what is it about a worm that makes it stimulate Th2 and a protozoa Th1? What is it chemically or physically about it that makes it go in either of those two directions? So that's a kind of fundamental question that you can use parasites to study. So, one of the first things that was done, and a lot of it here in the LPD, was to actually characterize the role of a lot of these cytokines produced by Th1 and Th2 cells in the response to these parasites. And interferon gamma and TNF were shown to be very important in intracellular control of parasite growth in infections like toxoplasma and leishmania, and of course IL-2 produced by Th1 cells is important for lymphocyte expansion and delayed hypersensitivity. A lot of work on the Th2 response, which is the most unique thing about parasites, and here IL-4 has been shown to be important in the generation of the IgE response, worm expulsion, IL-5 in helminth-induced eosinophilia, and IL-13 in worm expulsion, and important work by Tom Wynn here at the NIH in tissue fibrosis. So we could easily define what these cytokines were doing in terms of the response that we see, but there was a big question, and that was, is the function of these Th1 and Th2 cytokine responses to control a parasitic infection? And this is still kind of controversial, but I think this reflects my feeling and the feeling of a lot of people in the field, is that in the case of Th1 response, it's fairly clear-cut that the Th1 cytokines are protective. They are able to mediate killing of intracellular protozoa and are important for it. And of course, they perform a similar role with intracellular bacteria. The Th2 response is the one that's complicated and also quite interesting. Despite many work by many groups, including myself when I first started out in this field, it really is very hard to show direct helminth killing mechanisms that involve the Th2 response that are believable and hold up in vivo. But the Th2 response clearly has a major immunoregulatory roles and in infection, and it plays important roles in tissue homeostasis and repair. So if you consider the Th2 response, and this is a much more modern view of it. And in the old days, it was pretty much thought that all Th2 responses did was mediate allergy and helminth and resistance to helminths. And you can see now that, in fact, the Th2 response, even or Th2 cytokines, even at steady state, play a very important role in tissue homeostasis and all these different processes that are listed on the slide. And helminth infection is really only one of them. So, this raises the always fascinating question of where type 2 immunity came from. And if you go back and read textbooks, probably up till just a few years ago, it will say in the textbook that type 2 immunity evolved to control helminths. And I think that feeling is now that idea is now kind of fading away. And if anything, you might go in the opposite direction and suggest that the control of helminth infection is somehow a spin-off of these other functions of type 2 immunity in tissue homeostasis. In other words, its basic physiologic role rather than in host defense. So it's a fascinating question that is debated a lot in the field. So I'm going to now make a quick switch into Th1 and introduce a pathogen that stimulates the opposite type of response, which is Th1. And this pathogen is Toxoplasma gondii, which is worked on by a number of investigators at LPD. And what's so wonderful about this particular organism, it's not wonderful as a disease, but as a disease, it's a major opportunistic infection in HIV patients, and that's how we got involved in it from the beginning. But the fact that the type of immune response that it induces is so polarized towards Th1, and that's shown here. And basically what happens is early in this infection, there's a cytokine produced from innate cells called interleukin-12, which drives the production of interferon gamma, which is a host protective cytokine, and controls the growth of the parasite in host cells and mediates intracellular killing. And it also protects and helps maintain dormancy, because if the parasite does reactivate this or come out of its cyst form, which is its chronic stage, this Th1 response will quickly control the organism and keep it from reemerging. So it's and this is a very potent response, as you can see at the bottom here. You cannot detect any Th2 cytokines in this infection at all. It is so polarized off into the Th1 sphere. So when we were studying this back in the 90s, or first started studying it, there was something very interesting that we thought about, and that is that it was known that that interferon gamma can actually feed back on these IL-12 producing cells and actually generate more interferon gamma. So what you have here is a vicious loop where interferon gamma can induce more interferon gamma, and that response is almost certainly bad for the host. This is interferon gamma can in addition to controlling infection, can cause tissue damage and be lethal. And so a fellow at the time, Riccardo Gazzanelli, and I reasoned there must be something holding this response back. And a new cytokine had been just discovered, and Tom referred to it in his talk, and that's interleukin-10. And what interleukin-10 does is it suppresses cytokine responses. That was its original name, actually. And so we decided to look at mice that lack interferon, that lack IL-10. And just what I want to just point out to you very quickly is this is an actual experiment now for a change. So this is what happens when you infect mice, and for some reason I can't get the pointer to work here, but it's I don't know what I'm doing wrong. But anyway, oh, here, maybe that's it. Is that it? There we go. So if you infect wild-type mice, they can control the infection quite nicely. Interferon gamma knockout mice die because they need interferon gamma to survive. But these IL-10 knockout mice also died, and with almost the same kinetics as the interferon gamma knockout mice. And that was surprising to us. And it turned out that, in fact, the mice were dying from different reasons. The interferon gamma knockout mice had clearly more parasites and were dying of infection. In contrast, the IL-10 knockout mice had strongly elevated cytokine, a Th1 type cytokine responses that and there was evidence of tissue damage. So IL-10 was actually regulating the protective response, and this is illustrated in kind of a simple-minded fashion. I'll just ignore that here, and so normally you have both of these cytokines made, and the IL-12 helps in maintaining interferon gamma dependent host resistance. But if you get rid of IL-10, you actually have increased pathogen control, but also interferon gamma dependent immunopathology. And this may seem like a simple-minded kind of conclusion, but it's actually more profound, because what it means is that a cytokine, interferon gamma, which controls infection, is important. But IL-10, a cytokine that regulates that response, is equally important for the survival of the host. They're both highly protective cytokines, but they work in totally different fashion. Okay, so I'll just quickly go over this, and which is a question that we've been interested in for many years, and this is mostly the work of my colleague Dragana Jankovic, and that is, what makes this IL-10, and how is it regulated, and what is its trigger? And I'll just jump to the conclusion, and say that it is actually the Th1 cells themselves that are making the IL-10. So when you have an infection, you start out with making Th1 cells, and then you induce the production of IL-10, and IL-10 goes back and feeds back on the mechanisms involved in the generation of new Th1 cells. And in other work, we've uncovered another loop of this regulation of the Th1 response, which actually involves the neuroendocrine system, and both of these mechanisms play an important role in dampening the this very potent response to this infection in the host. Okay, so finally, we're going to switch to the last question here, which is to go back to the beginning and really ask, what is it that drives these different types of, or these two very distinct outcomes? And this has been probably one of the most fascinating questions in the field. And what we and many others believe is that what this involves is signals delivered to precursor T helper cells, these are called THPs, that drive them in one direction or another. So what are these signals and where do they come from? So it's the general feeling is that there are what we call accessory cells that recognize different microorganisms, and here's a bunch of them listed in this in the pot in the center here. And so the idea would be that these cells have receptors or are somehow triggered by different pathogens, and they make different factors that drive the production of all these different T cell subsets that we were talking about. So this is an important idea in the field for effector choice. But we decided to focus on one particular cell, and that's an important cell called the dendritic cell. And what's important about dendritic cells, as you may know, is that they play a basic role in the immune response anyway. They're very important in presenting antigen to generate T cell responses, period, regardless of whether they're Th1 or Th2. So if you could somehow stimulate a dendritic cell to make a signal, you could not only present the antigen, but you could also dictate the effector choice in one direction or another. And this is what we've been studying. And here we had a really a neat system, because unlike many other organisms, the two parasites that we studied in the lab, which is toxo, which I just talked about, and schisto, which is a helminth infection, you can actually grind up these organisms and get out a soluble extract that will stimulate Th1 or Th2. You don't even need the organisms themselves. So we're in a situation to biochemically characterize what it was in the organisms that makes them go in one direction or another. And I'm just going to summarize years of work in terms of what these molecules are and how they work in particular on dendritic cells. And that's shown here. So here's the toxo molecule. And this was work of Felix Yarovinsky, a postdoc in our lab. And he purified the actual component in the soluble extract of toxo. And it turned out to be a structural protein of all things called profilin. And in the process of identifying the ligand, he also identified its receptor or receptors on dendritic cells, which is was two newly discovered toll-like receptors called toll 11 and 12, which signal through an adapter molecule called MYD8 and stimulate the production of IL-12. And IL-12 is what drives T helper precursor cells into Th1 cells. So this was pretty straightforward, and profilin is a very, very potent molecule, at least in the mouse, and is able to stimulate. It's almost like LPS in its ability to stimulate strong immune responses. So the Th1 side seemed pretty straightforward. Th2 side, however, is complicated. But that being said, we were able to identify the molecule, the major molecule in this schistosome egg extract. And interestingly, this is a totally different molecule. This is a T2 ribonuclease called omega-1. And this just shows its purification. And to cut a very long story short, omega-1, which is shown here, stimulates dendritic cells in a very, very different way than profilin. So profilin highly activates dendritic cells. You get a strong T cell activation signal. In contrast, omega-1 gives you a very weak activation signal, and in certain cases even looks like it's inhibiting dendritic cell function. And that weak T cell activation signal is associated with the generation of a Th2 response. So this is how we think effector choice is working in a model helminth and protozoan system. So I'm going to end and come back to the very beginning of all of this, and pose a question that kind of haunts me, probably more than it haunts Tom, and that's shown here. And so we've had this enormous interest in the immunology of parasites, and this is actually a quote from a famous Brazilian pathologist, Zilton Andrade, who actually used, this was in a schistosome meeting. He said, has schistosomiasis done more for immunology than immunology has done for schistosomiasis? and he raised a very, very good point, because we really still don't have major vaccines for parasitic infection, except for one, which I promised to come back and tell you about, and that is a vaccine called RTSS. And it has about the worst name for a biological I've ever seen, Muscorix, which is supposed to remind you, I guess, of mosquitoes and malaria and all that. But so what it is if you remember the Nuissensweigs and their discovery of this CS molecule, this is what happened to it. So in terms of vaccine engineering, they took this central repeat that I showed you, which is what the monoclonal antibody recognized. They tacked some T cell epitopes onto it, and they genetically engineered this into the surface antigen of hepatitis B virus as both a carrier and a way to I think to target it into the liver, which is where this particular stage of malaria takes place. And so the initials are where the RTSS comes from, and so what I want to say is that this construct was made around 1980, 1982, and it was studied by the Army, and it didn't really go anywhere. There was vaccine trials done with it, but then someone happened to put the right adjuvant together with this molecule, and suddenly they started getting promising results. And that adjuvant is something called AS01. It's made by GlaxoSmithKline, and it's still, to me, pretty much voodoo. It's a liposomal formulation of monophospholipid A, which is a known adjuvant, and a saponin fraction from a Chilean soapbark tree. But anyway, this works, and it works not particularly well, as I'm about to describe. And so the idea, and it was just described to me today in a conversation I had with a malaria vaccine guy, is that you have your RTSS and you have your adjuvant, and you mix them at the time of the vaccination, and this has been studied now for at least about 15 years of vaccine trials. And this is where they are now. They have a 3-dose schedule, and this was, I think, published in, I think it was Lancet a couple in 2 or 3 years ago. So there's a 3-dose schedule of vaccine, and you can imagine coming back and vaccinating people 3 times, and they've added a fourth boost, which seems to be very important in getting significant protection, and that boost gives you a 36% reduction in clinical malaria in children aged 5 to 17 months and 26% in infants. So this isn't really much. The way it's been described to me is you have 198 million cases of episodes of clinical malaria, and you can eliminate 30 million of them with this. And I think the key thing is we really still don't understand how this vaccine works. It seems to be associated with antibody, but there's evidence in animal models that CD8 T cells are important. And the only recent clue is that the booster seems to be very important, the last booster. And if you lower that boost, it can play a major role in the outcome by increasing antibody affinity, is the one thing that they've been able to measure. So all this work that I've been telling you over all these years, and great neglected diseases, has given us this. And you can just see, I see some young people out in the audience that I know, that this is what an enormous challenge is. This field is still sitting there waiting for us to understand how to move forward and generate important forms of immunologic intervention. I should also mention that these results, I forgot to mention it, were in the context of regular malaria prevention. So they're bed nets, everything. So you're looking at the combined effect of a vaccine upon with everything else. But anyway, they're going ahead with it, and there's going to be a trial with a much larger trial that GSK is sponsoring in 3 different African countries, and just to see if they can, again, that if just by helping those 30 million cases, whether this is going to make a significant impact on malaria itself. So you can see the challenge that remains in this field, and this is a statement of it. It's really, I think we still need to understand more about, a lot more about the effector mechanisms that mediate host resistance to parasites. And we need biomarkers for these protective responses in humans. That's really important to know. In almost any human infectious disease vaccine situation, we need ways to find out whether a response is protective or not without exposing someone to the disease. And then also, I think those of us who work on mouse models and other models really need to be thinking more about how to translate them into actual translational interventions. So I'll end there, and this is just a huge list of people that have worked with us over the years on the work I'm describing, and I'm very fortunate to be here at the NIH and to be in LPD, which is a great place to work and with great colleagues, and we've had very wonderful external collaborators over the years that are also mentioned here. I'd also like to acknowledge Bill Paul, who is a very important colleague in a lot of the immunologic work that we did, and unfortunately is no longer with us. So I'll stop there, and if there's any questions, I think we're right on the nose, 6 o'clock.
We don't take advantages of, it's not a very nice talker. We don't take advantage of some of the advances that happen there. For instance, in the cestode parasites, which were briefly alluded to, there are vaccines, and these vaccines are 100% effective, and they stand out as very different than a lot of the helmet problems that we have in immunology. And you almost can't get anyone to work on it and to apply it. So there's a lot of favoritism for what is worked on and what is not.
Yeah, I forgot to mention there's a whole world of veterinary vaccines that where there has been more progress, and that's just simply can be credited to the fact that it's easier to do trials and to test hypotheses in animals than in humans. So, but what Ted is alluding to is the fact that in some parasites it's not so difficult to immunize, but they're not ones that that are like the big guys that some of the big guys that were mentioned here, big diseases. He may differ on that. I don't know.
So the idea that a parasite, I'm curious about the relationship of eosinophilia to parasites which invade tissue as distinguished from those that don't. Now, many years ago, we were taught in medical school that was an important finding. Is that true anymore?
Well, I think Tom is a better person to answer that than I. I mean, I think it is a response to tissue invasion for sure. Although, as I was trying to point out, eosinophils occur in other situations, but I think I think the idea of tissue damage is also a very important thing in the Th2 response. Remember, the Th2 response is now thought to be a healing process, a tissue repair process. Say what? I'm sorry. Yeah. Yeah, for the other people, yeah. So it's thought to be the Th2 responses I showed you on one of the slides is meant to involve tissue repair. That may be its real function. And helminths are great at tearing up tissue. That's one of the things they do. You experience them tearing up your tissue. So I think what you just said is probably still thought to be the case, and Tom can clarify that.
No, I completely agree. I think you have to think about two different things. So what drives eosinophilia in the blood is largely we know is IL-5, and it acts as a hormone essentially on precursor cells in the bone marrow and then drives them in the blood. But eosinophils are really tissue resident cells, and in humans at least you can't really study that. You pretty much have access only to the peripheral blood compartment, and I think largely all the tissue damage done by eosinophils happen in situ, and so I think one is the regulation of eosinophilia, which I think we understand a lot about. It's really the rest of that, and what's probably driving even that is tissue invasion, because those organisms that don't invade the tissue really don't drive eosinophils.
But I just wanted to I made the point, I just want to say it again, in just general point, is how important helminths have become for studying all of these Th2-associated responses. I mean, my starting point was that, the study of the immune response to parasites has taught us a lot about immunology. And one of the big things, the unique things, is the Th2 response, because it really doesn't exist in any substantial way to any other type of organism. So we've learned a lot from studying this. But we're left with this fascinating idea about who came first, the parasites and the need to control them, or just the need to repair and maintain tissue homeostasis.
The roundworm has been a stalwart in the study of aging, development, genetics, and it's all based on the principle that you can feed the worm a bacterium which contains whatever it is you want to put in that's going to permit you to identify and study it. So that sort of prompts something in my head about the relationship, if any, between the bacterial microbiome and the parasitic infection of the intestinal tract. Is there sort of a yin-yang there, or is this are these two separate worlds?
I think Tom can speak to that better than I.
Well, I think it's clear that that there is an effect of probably a two-way effect of some of these intestinal worms and the composition of the microbiome, and probably there is some influence, we have to assume, in the other direction as well. That is, some of these bacteria may in fact influence the growth and development of a particular parasite. I think what's less clear at the moment, at least, are these what turn out to be relatively subtle changes in the microbiome induced by even heavily infected, parasitized individuals? Does that really actually alter in any way the immune homeostasis sort of systemically? And I think that's sort of the next hurrah, in a sense, for how people are going to begin to understand these sort of complex interrelationships between host tissue, host microbiome, and these external pathogens.
So the other wrinkle to this, what you're asking is, and maybe Tom was referring to this, is you have to think of parasites themselves as commensals, just like there may be a wormiome, or we know that there are protozoa that are very important commensals now that have been described. And then there's also this whole fascinating idea of the hygiene hypothesis that, that we've carried these around for years, and they're part of our immunologic makeup. And of course, we stop having parasitic infections or worm infections, we completely change our immune responses, and that could be in a bad direction. So that's another fascinating aspect of which you need to get Tom back to talk to you about another time.
Well, I want to thank you both, and thank you very much for coming and sharing your extraordinary experience, which I suspect that many of us, at least me, had never seen or heard before. So it was very exciting. So thank you all very much. And we hope we'll see you next week when it'll be diabetes and artificial sweeteners. Maybe they affect the helminths too.
It all has to do with helminths.
Thank you very much.